Fatal diseases such as chronic wasting disease are caused by prions, long chains of proteins that act differently depending on how they fold themselves. A team led by Dr. Bruce Chesebro at Hamilton's Rocky Mountain Laboratories has observed early establishment of such a disease in mice. I talked to Dr. Chesebro about how prions act. If a prion looks one way and then all of a sudden it folds itself, it has a whole different effect.
Well, that idea is common to all proteins in our bodies. All proteins that we eat in plants and animals, and all proteins in our bodies. Proteins aren't just a long amorphous string of amino acids that looks like a strand of spaghetti. They are always folded up, for the most part, into a very precise structure. Their folded structure, that is totally important for their function. It allows them to form pockets where they can trap molecules and make certain chemical reactions happen faster because they get all the components of the reaction attached to the protein. It makes them react together really fast, and that's how enzymes work to make all of our reactions in our body. And so the whole thing of protein structure leading to function, good function, not bad function, is well known and established, and it's a very interesting concept. Not so well known to the general public. And when you do things like cook eggs, you denature the protein, which is essentially you change it from its normal folded structure into a different kind of folded structure that doesn't look anything like its folded structure, and that's what you get when you get hard-boiled eggs.
Now, prion diseases that we're familiar with include chronic wasting disease, actually Alzheimer's and some of that sort of thing. How does this affect those studies?
Chronic wasting disease is definitely a disease involving prion protein, so it's totally directly related to what we're doing. Alzheimer's disease and Parkinson's disease involve the misfolding and aggregation of different proteins than prion protein.
Oh.
They involve misfolding of a protein called tau in one case, or a protein called alpha-synuclein in another case. That's for Parkinson's. And AAP is the protein for Alzheimer's, and a little piece of that called A-beta misfolds and makes the misfolded aggregates in Alzheimer's disease. So those three diseases don't involve prion protein directly as much as we know. All in all, some aspects of those diseases, because of their prion-like phenomena, are going to be similar to what we see, and some are going to be different. And we don't really know if what we saw on these blood vessels is going to be seen in the other diseases, and we haven't studied that yet. But other people will probably study that.
In the study, the scientists injected infectious scrapie prion protein into the brains of mice.
Yeah, that's a sheep disease that's been around for 300 years in sheep almost everywhere that they're sheep. They're scrapie, except for they've pretty much eliminated it in Australia and New Zealand. It sounds a little weird. It's a sheep disease, but it's been transferred to mice and hamsters, and people all over the world work with it in mice and hamsters, which are a lot easier to work with in large numbers than sheep, obviously.
Now, you injected scrapie into some mice and actually saw some things that you weren't expecting.
That's right. Other people showed that the infectious part of scrapie appears to be this misfolded form of prion protein. And we know how to stain for prion protein, and we have ways of distinguishing the normal form from the misfolded form. And we were actually able to see evidence that of the material that we'd inoculated within 30 minutes of inoculating it, which is, nobody's ever looked that early to find it. It's really a surprise to us, and it inspired us to go on and see how things progressed over the next few days or weeks. And to our great surprise, we could see that the amount of the abnormal prion protein increased pretty obviously at 7 days, and in some mice, we even saw it increased at 3 days. And from 7 days on, it increased more and more and more. And that's pretty remarkable when you have a disease that usually takes a half a year to kill the mice. We think we can use this tool as a tool to study how to block the spread at early times when the mouse isn't completely overwhelmed with the infection.
Now, in your article, you mentioned that it also attaches itself to the blood vessels. Is that right?
Yeah, that was a pretty big surprise. We think that's just part of the normal clearance process of aggregated particles that get into the brain that are formed in the brain normally in different kinds of metabolites, or the remnants of dying cells have to be cleared from.
Mm-hmm.
They are cleared through a flow system where fluid flows along the outside of blood vessels. It's a bizarre system that it goes that way, but apparently our material follows that pattern, and it seems as if the brain is trying to get rid of it. It does get rid of it, but in the mice that are normal mice, they get rid of the stuff that we inoculate, but at the same time they're getting rid of the material we inoculated, they're also replicating it. They're using a small amount of the stuff, of the inoculated PRP, to serve as a template, we think, to form new aggregates of abnormal form. And this is sort of a cyclical phenomenon.
I'm assuming that you're following that path now to watch how that works.
Exactly. We're trying to figure out where it happens, how it can spread from the place where we inoculated it to other places. We're currently following it to the cerebral cortex and to the thalamus and to some other places in the brainstem. And we're also, at the same time, we're trying to figure out in more detail what kind of brain structures is it attached to when it's moving around and when it's sort of accumulating. We think it moves around by following nerves within the brain, the actual nerve fibers. But we think it accumulates in very special places. And this isn't part of this paper, but we think that different strains of scrapie may accumulate in different kinds of cells. Some accumulate in cells called astroglia, and some accumulate more in the nerve cells. And some can do both, whether that has some important implications for what kind of disease you get and what kind of drugs you might use to block the spread. The normal prion protein has a DNA copy, and it's made into RNA and then made into the normal prion protein. But we're not really affecting that process by infection with our abnormal form. We're just using some of that normal protein and getting it to fold differently into a misfolded version that turns into aggregate.
And I'm assuming that you've replicated that in a number of different mice.
Yeah, we have. It may work best with one. We've tried it with several different strains also. And right now, that early replication is more prominent with one particular strain that seems to be a stronger strain. We have it at a higher titer. And maybe it replicates faster, too. We don't really know for sure. But this super early replication may be a reflection of the speed of this one strain, and some of the other strains appear to be slower. But we're trying to follow that up and understand why that is also.
As far as what you and your team or other folks in your lab are going to be doing at this point, what next step are you looking at as far as understanding this study?
Well, we're trying to pin down the differences among the scrapie strains that we have here, and see whether this helps us better to understand why the strains can go to different tissues and cell types. And we're trying to pin down more accurately whether we can block this. Using this technique, we think we can intervene with drugs and perhaps block some of these earlier steps more easily than when you're starting with an animal that has a widespread infection. And then we're also thinking that since we can follow it so closely early, if we see a partial block, we'll notice it more looking earlier than if it's only a partial block later, we might not even notice it. So we think it'll give us an added sensitivity that will allow us to refine things and improve them.
What do you use to actually see this happen?
Well, it's actually pretty conventional microscopy. The real trick to doing this is to have a micro-injecting system that puts a very tiny needle in the brain in exactly the same place every time on every mouse. So we anesthetize the mouse and put it in a special apparatus that holds its head in a certain position, and then using micro manipulators, we can arrange to have the needle hole in the skull go in exactly the same place in every mouse and go to the exact same depth. And then we have a pump that pumps the injection in. We don't just inject it by hand. And the pump pumps 1/2 a microliter of fluid in every minute. So this is incredibly slow. There's no way we could do this by hand. Mm-hmm. And it restricts the distribution of the material we put into the brain into a very small pocket within the brain. And then that takes a few minutes to set it up and then do it. But that's the trick. And then when you look either 30 minutes later or 3 days later or 30 days later, whatever, and take the brain out, standard pathology techniques, putting it into paraffin blocks, and we section the brain, and we search for the sections where the needle track was. And then when we find the area, then we have a whole lot of sections that are adjacent to that we study with special stains. But that's all conventional microscopy. The trick is having the microinoculation be very, very small, so it starts in a single place, and then you can find that place and look at it.
Yeah, a number of people helping you with this, and of course a team always works on all these things. And
we have a good team. Brent Race and Katie Phillips are the ones who have been doing the micro-injections, Dr. Rangel. And then we have a number of other people who have helped us with various aspects that are co-authors on the paper.
I guess one of the questions, taking a look at a general look at the body and a mouse body, a human body, why does the prion disease affect just the brain or the spinal cord or that area?
Well, that's a really good question, because prion protein is found in many tissues and many different cell types throughout the body. And even the aggregated form of prion protein in scrapie-infected sheep or CWD-infected deer or elk can occur outside the brain. And the same is true for people who have Creutzfeldt-Jakob disease. They can have accumulations in other tissue, although it's less frequent than in some of these animal models. But we don't know whether these accumulations of the abnormal protein in other tissues is damaging. I think there is evidence that in some cases it's damaging, and in some cases the damage isn't so obvious. We can see that the organs don't look normal because they accumulate these masses of abnormal protein, but we can't put our finger on what the functional abnormalities are.
The research was conducted at Rocky Mountain Laboratories, an NIAID facility of the National Institutes of Health. I'm Steve Fullerton of KLYQ, 1240 AM, Hamilton.